Hi everyone, and welcome to the first lecture in a two-part lesson on Chapter 14, DNA Structure and Function. So I present to you a model of DNA. Looks pretty complicated, doesn't it? Well, we're going to break it apart, we're going to simplify everything, and you're going to learn all the details of DNA in this chapter. So Gregor Mendel, remember him? The Austrian monk called them discrete factors in the 19th century. He didn't know anything about genes, but he knew there was some substance, some essence that allowed the physical traits of a parental plant to be transferred to a progeny plant. We know them today, of course, as genes, and you know already from your studies that genes are made of DNA and various proteins. You know that genes are carried on structures called chromosomes. But just what is DNA, and why is DNA the repository of genetic information? In other words, how does the information, the instructions for flower color and seed shape, or for that matter your hair color, your eye color, 5 fingers on each hand, height, stored in DNA, and how is that information passed from generation to generation? That is the subject of this chapter. So our objectives here are to describe the Watson-Crick model of DNA, describe Rosalind Franklin's role in the discovery of the double helix, explain the significance of Chargaff's rules to the structure of DNA, explain what is meant by complementary base pairing, describe the process of DNA replication, differentiate the conservative and semi-conservative models, describe the action of DNA polymerase during replication, compare and contrast replication in prokaryotes and eukaryotes, lots of similarities but one basic difference, and describe the different forms of DNA repair. So I showed you this illustration back in Chapter 10 of the cell cycle, and I just want to help you realize where we are in this whole cycle that we call biology. Okay, so we talked about metaphase, and you know all about the steps of metaphase, and then before the cell divides, right, goes into the mitotic process, goes to this G1 or this growth phase. And then it goes into this S phase, and you'll remember that S stands for synthesis. And this is where DNA replication occurs. Okay, so I don't want you to forget about the cell cycle. I want you to try and tie the pieces together from chapter to chapter to chapter. And the replication or the duplication of DNA, as we'll see in Chapter 14, occurs here in the S phase of the cell cycle. Now, I've shown you this illustration before, and we'll learn more about it in Chapter 15. And you'll recall the central dogma just describes the transfer of information in one direction from double-stranded DNA to single-stranded RNA and ultimately to protein. And this is really where we want to get to, because everything that you are and everything that you think and what you look like, all of that is derived from protein. Okay, but the S phase of the cell cycle is replication. Now, the central dogma is transcription of DNA to RNA and translation of RNA to protein. But back in the S phase of the cell cycle, we have replication. And that's where you take DNA and you copy the DNA. And you make 2 copies, right? Because we have to eventually make 2 homologous chromosomes before we can divide, if we're a cell. Enter DNA. Friedrich Michener, Swiss chemist, discovered DNA in 1869. So we've known about DNA for a long, long time. He extracted a white substance from the nuclei of human cells, and he called this substance nuclein, because he got it from the nucleus, Nuclein, being somewhat acidic, then became known as nucleic acid. And so you learned back in Chapter 3, I think, that DNA and RNA are nucleic acids, one of the macromolecules that you learned about. At the time, while the former structure of DNA was unknown, the components of DNA were known, and the components are a 5-carbon sugar. That sugar is deoxyribose. Remember, it's oxyribose in RNA, or rather ribose in RNA. Deoxyribose in DNA, a phosphate group, you know that as a PO4, and a nitrogenous base. Nitrogenous just means nitrogen containing, and you know, you've learned this already, that the bases in DNA are adenine, capital A, thymine, capital T, guanine, capital G, and cytosine, capital C. Now, in this little illustration over here is a picture of my DNA. When I do the lab for this class live, I have my students extract their own DNA, and you may have done this in high school or 7th grade life science, where you smashed up some strawberries or smashed up a banana and you extracted the DNA from there using salt water and a little dishwashing detergent. Well, I have my students extracting DNA from their own cells. We just take a toothpick and we just gently scrape the inside of our cheeks, and we extract the DNA from those cheek cells. And I did this, I don't know, maybe 20 years ago. I still have it. I did this maybe 20 years ago. And that white substance that you see in there is my DNA. Stored in a little bit of alcohol in this little microcentrifuge tube. And the interesting thing to me, I've always found this fascinating. Even when I was working, my graduate research was in frog genetics, and I had to take a lot of frog tissue and extract the DNA. And it's interesting to me, right? DNA is in your cells. Not only that, it's in the nucleus of your cells. And your cells are microscopic, and the nuclei are microscopic. So until the chromatin condenses, you cannot see chromosomes. So in other words, you can't even see DNA. It's submicroscopic before it condenses at that point. And yet you can pull this stuff out of your cells, and you can visibly see it with the naked eye. This is not enlarged at all, and you can actually hold it in your hand. The DNA from your cells, which fits inside of the nuclei of your cells. That to me is incredible. Anyway, I love to show that picture because it allows you to see the fact that you can see DNA, which is inside the nucleus of your cells as we speak. So here's the structure of DNA and RNA, of course. But we're thinking mainly DNA here, right? So here we see the pentose sugar. Pentose just means 5-sided, pentagon, right? 1, 2, 3, 4, 5-sided sugar. Now, if there is an oxygen, or really an OH group down here, then that's ribose sugar. But when you take off the oxygen, you deoxygenate the sugar, then it becomes deoxyribose sugar, and that's what we're talking about with DNA. So we have a pentose sugar. Here's your phosphate group, the phosphorus with 4 oxygens attached. And here's one example of a nitrogenous base. Nitrogenous base because it contains a lot of nitrogen, as you can see. Now this nitrogenous base can have a double ring. There's 2 examples of that. We call those purines in general. Or it can have a single ring, and you see 3 examples of that, but really we're looking at these 2 for the moment. And we call those pyrimidines. If it's a single ring nitrogenous base, it's a pyrimidine. If it's a double ring, it's a purine. And you can see we give them names. Here is adenine. And these are just amino acids. Here's adenine and guanine. Those are your 2 purines. And here's cytosine and thymine. Those are your 2 pyrimidines. So phosphodiester bonds are the chemical bonds that link nucleotide monomers. And you know this because we built strands of nucleotides back in Chapter 3, and we learned that phosphodiester bonds, by way of dehydration synthesis, right, we form those bonds, and it's phosphodiester because we're connecting this ester bond between the phosphorus. Okay, the bond is between the oxygen here on this pentose sugar and the phosphate, so it's the 3' O and the 5' P from this pentose, and that's where we form that phosphodiester bond. It's called diester because you have an oxygen bound, a bound oxygen on either side, diester. So the phosphodiester bonds form between the 3' and 5' carbons of adjacent nucleotides. So this is a nucleotide, base sugar phosphate, and this is a nucleotide, a different one, base sugar phosphate. And there are many thousand of nucleotides connected to form long nucleic acid polymers, polypeptides eventually, right? And so nucleic acid polymers are intrinsically polar, and that means you have a 5' end and a 3' end, and so you have this phosphate on the 5' end of the strand and this hydroxyl group on the 3' end of this strand. And the sequence is always written 5' to 3', okay? And this will become more, I think, comprehensible when we look at a longer strand. So, and here we see a longer strand. So we have phosphate, sugar, alternating, phosphate, sugar. And this is very strong. There's a covalent bonds between them. So they're very strong, and it forms quite a rigid, relatively speaking, quite a rigid structure to which the nitrogenous bases are attached. And so because it's semi-rigid, I should say, because it does fold and twist, but because it's semi-rigid, we call this the phosphate sugar backbone, because it's alternating phosphate sugar all the way down. You can have 1,000 nucleotides here, and you're going to have that alternating phosphate sugar backbone. And on the 5' end, see the 5' carbon has this phosphate group attached, and on the 3' end we have this hydroxyl group attached. Alright, so 5' to 3', phosphate to hydroxyl group. Let's go to the video. Okay, this is by MITx Bio, the structure of DNA. It's a good video. Kind of goes a little bit fast. You can pause it and replay it as many times as you want. But it gives you a basic general idea of the structure of DNA. Now, Watson and Crick are the two scientists that ultimately published the structure of DNA, that is the double helical structure of DNA. Now, you learned about the double helix back in one of the earlier chapters, and we'll see it in more detail here. But again, they are credited with deducing the double helical structure of DNA. That was in 1953. Francis Crick was Englishman, James Watson was an American. And they used the experimental results of others, including Chargaff and including Rosalind Franklin, to deduce the double helical structure. And that granted them, or that earned them, the 1962 Nobel Prize. Now, not to be forgotten, because often she is, Rosalind Franklin was a British chemist, and her specialty was X-ray crystallography. And she worked with a lab assistant, and they took these X-ray photographs of the structure of DNA. And Watson and Crick used her photographs to help them delineate the double helical structure of DNA. Now, Watson and Crick got the Nobel Prize, but if it was not for the work of Rosalind Franklin, I'm not saying the double helical structure never would have been ascertained, but it would have taken them much, much longer. It was her photographs her X-ray photographs that confirmed the helical structure of DNA. And so as a result of that, some years ago, for a celebration of women in science, I did a short video on Rosalind Franklin. And if you get to watch that, you can. Here's the link, and the video is in the embedded videos, on the embedded videos page. Any questions? Easy enough, right, to get us started in Chapter 14, DNA structure and function. Let me know. That's the end of Part 1, and I will see you soon.